We're here at the European Retina Meeting in Brighton, just down the road from Uckfield headquarters for Scientifica, and we're with Dr. Greg Schwartz from Northwestern University in Chicago. Um, Thank you for joining us. How are you doing today?
Great.
Great. Okay. Your lab focuses on the circuit mechanisms underlying sensory computation, particularly within the retina. What are you hoping to understand about these circuits and why?
So the retina ends up being one of the best model systems both for sensory computation and in general for mapping and tracing neural circuits in the brain. In large part, there's two main reasons for this. One is that we understand the cell types better in the retina than perhaps anywhere else in the nervous system. There are over 100 different types of cells in the retina, and we have molecular markers and functional characteristics of many, many of them. And the second reason is we have precise control over the input, and we have precise measurements of the full output of the retina, right? So we can stimulate with light, which is of course the natural input, and we can record the spikes of the retinal ganglion cells, which are the complete output of the retina. So those two things combined make it a really spectacular system in which to trace the way information flows through a full neural circuit.
Okay, and what are you currently working on in your lab?
So among those cell types, there are still many that are not completely characterized, especially functionally, and the next real revolution is going to come from not only knowing all the cell types, but knowing their connectivity. And there we're much further behind. So my lab is trying to map synaptic connectivity between the different kinds of interneurons and the retinal ganglion cell outputs to understand the functional computations within the retina. And we're using both physiology and imaging techniques.
Okay, and where are you hoping to get to? Where's your final goal for your research?
So there's a number of goals. I mean, it's always going to be an ongoing process, but I think the ultimate test of when you understand a neural circuit is when you can model the full circuit from the light input to the retinal ganglion cell output in this case, and not only understand how it works, but why, which is always the harder characteristic is the why part. But by why, I mean what function does it have in the visual perception of the animal. So mouse genetics is starting to get us there, where we can manipulate precisely specific circuits and then see the functional output in the behavior of the animal. So that's really the ultimate goal. Understand how synapses do their thing and why that makes the animal have a particular perception or behavior.
Okay, and what techniques do you do to do this?
So we use primarily electrophysiology and imaging, both functional imaging and anatomical imaging. So more specifically, we record the retinal ganglion cells, both their spikes and their synaptic currents. And to understand connectivity, we do both anatomical imaging experiments to look for synapses, and we also do paired recordings to directly look for synaptic connectivity between a presynaptic and a postsynaptic neuron.
And you use Scientifica's Multiphoton imaging system as one of your tools for doing this. How does that help you to attain your research goals?
The multiphoton imaging system is really critical for our work in the retina because, again, with mouse genetics, we are able to target specific cell types by expression of fluorescent proteins. And while this is done in confocal microscopy in many other parts of the brain, we study functional light responses in the dark. So confocal microscopy is not feasible to do this in the retina. We bleach all the photoreceptors and there's no light response anymore. So we need to do this under infrared illumination to photon to be able to target our cells. So that's what we do every day to be able to find specific cell types.
And how have Scientifica as a company helped you in particular?
Scientific has been really great because these are very custom systems. We use infrared illumination, we use visual stimulation through parts of the microscope that are not normally used for stimulation. We stimulate with spatiotemporal patterns through the condenser, and all these custom things were worked out in close collaboration with the members of the Scientifica team. And their transparency in telling us about how the light path works and the scope and allowing us to tinker with it has really been fantastic. And on the customer service side, when something goes wrong or something needs to be tweaked on the scope, the representatives that come to my lab know where every screw and bolt in the system is, which really makes a huge difference and is not characteristic of the larger imaging or electrophysiology kind of companies. Sometimes we as end users feel like we're kind of small fish in a big pond, but with Scientifica we really do feel like we're part of the team.